I am Pratiksha Prabhakar Kare. I am studying in the B Pharmacy second year. Today I am going to give a seminar on the simple staining. Staining. Staining is a technique used to enhance a contrast in the microscopic images. Due to this staining technique, we can observe the bacteria or this cell clearly. We can observe its size, shape, its color, its morphological arrangements. Stain. Stain is an organic compound containing the benzene ring. It is used to color the cells or the background. There are two types of stain, that is acidic stain and basic stains. Acidic stains are negatively charged acid radicals, while as the basic stains are positively charged basic radical. Acidic stains imparts color to the background and basic stain imparts color to the cell. Examples of the acidic stain are eosin and nigrosin, while as the examples of the basic stains are methylene blue, crystal violet, and safranin. Simple staining. Simple staining is a staining that uses only a single dye. In the simple staining, everything is stained with the same color. Simple stains are used to stain the whole cells or to stain the specific cellular component. In this simple staining, we can stain the whole bacterial cells, or we can stain the only the specific cellular component that which we want to stain. That is, if we have to stain only the nucleus, then we can also stain this with the help of simple staining. There are two types of simple staining, monochrome staining and negative staining. Monochrome staining is also called as the positive staining or direct staining, while as negative staining is also called as the indirect staining. Monochrome staining. Monochrome staining is a staining technique that stains the bacterial cell in a single color. This monochrome staining stains the bacterial cell in single color. Basic stains carries a positive charge and bacterial nucleic acid and certain cell wall component carries the negative charge. So that when we stain the bacterial cell with basic dye carrying a positive charge and cell carrying a negative charge, opposite colors get attract towards each other and the cell get stained. But the question arises that why the bacterial cells carries a negative charge? The bacterial cells carries a negative charge because of large amount of carboxylic group located on the cell surfaces due to the acidic amino acids. Therefore, when the ionization of carboxyl group take place, it imparts a negative charge to the cell surface. What is the procedure of monochrome staining? We see that the procedures of monochrome staining. Firstly, we have to make a thin smear on the clean glass slide. Then we have to heat fix the smear by passing the slide 2 to 3 times gently over the Bunsen burner flame with the smear side up. Flood the smear with the methylene blue. Allow it to stand for 2 to 3 minutes. Wash the stained smear with water and air dry it. Flood the smear with methylene blue. Allow it to stand for 2 to 3 minutes. Then wash the stained smear with water and air dry it. Then we have to observe this smear under the oil immersion objective. Here we can say that how we have to prepare the smear. Firstly, we have to take a clean glass slide. We have to make this slide grease free. Then we have to add a drop of distilled water. Here they are adding the drop of distilled water directly by the bottle, but we have to take this drop of distilled water by the nichrome wire loop. Then we have to add the inoculum inside this water drop with the help of nichrome wire loop, and we have to prepare a thin smear. Uh, then we have to heat fix this smear by gently passing the slide 2 to 3 times over the Bunsen's burner flame, and then we have to air dry it. After the air dried of the slide, we have to flood this smear with this stain. In monochrome staining, we are using the methylene blue stain, so that we have to pour the 2 to 3 drops of methylene blue stains on this smear and wait for 2 to 3 minutes. After 2 to 3 minutes, we have to wash this stain with the tap water gently by pouring 2 to 3 drops of the tap water over the slides. Then after washing the slide, we have to air dry it, and after air drying, we have to observe this slide under the oil immersion objective under the microscopes. Observation: in the monochrome staining, we get a colored cells against the in this we get a colored cells. Here we can see that a blue color cells we can observe here, and background remains the colorless. Negative staining. Negative staining is a staining in which the bacterial cell is not stained directly. In this staining method, the background gets stained and cell remains colorless. Negative staining requires the acidic dye. In monochrome staining, we uses the basic dye, and here in the negative staining, we requires the acidic dye. Examples of negative staining, negative acidic dyes are nigrosin and India ink. The acidic stain readily gives up the hydrogen ion, and the chromophores of the dye becomes negatively charged. Since the surface of most of the bacterial cell is negatively charged, the cell surface repels the stain, so that cell remains colorless and background gets colored. Procedure of negative staining: place a small drop of nigrosin close to one end of clean slide. Then place the slide against a drop of suspended organisms at 45-degree angle, and allow the drop to spread along the edges of applied sites as shown in the figure. Then using a aseptic technique, place a loopful of inoculum from the bacterial culture in the drop of nigrosin and mix it well. Then push the slide away from the drop of suspended organism to form a thin smear and air dry it. Examine the slides under the oil immersion objective. Here we get the results, that is negatively stained cocci cells. Here we can say that the colorless cocci spherical shells are present against the colored background. Thank you.